Hey everyone, welcome back to my channel. So today I'll be talking about DNA transcription and specifically the initiation and elongation process of transcription. So transcription is basically copying the genetic information which is stored in the DNA molecule and using that information to synthesize RNA, and then RNA eventually is used to synthesize proteins, and proteins are important to perform cellular function, as well as they also define the cellular identity, and cell eventually makes up the organ, and organ makes up the body. So this is an important chain of life. So there are basically three steps of transcription: initiation, elongation, and termination. So I just want to quickly go through the difference between the replication and the transcription. So replication, which uses DNA polymerase enzyme to synthesize new DNA strand, whereas transcription, it involves RNA polymerase enzyme. Replication uses deoxyribonucleotides, whereas transcription uses ribonucleotides to synthesize new strand. Replication needs RNA primer and transcription does not require RNA primer, which is one very important difference. And replication, where the new strands are formed, they remain base paired, whereas in transcription, the new strand which is formed, it does not form or it does not remain base paired with the template strand. Replication, the important difference is the replication copies the entire genome, whereas transcription only copies the certain parts of the genome. Replication is very accurate because the entire genome is then going to pass on to next generation. So therefore the replication process has to be very accurate, whereas transcription is less accurate. And lastly, in replication, the enzyme also performs the proofreading, and therefore the process is also very accurate, whereas in transcription, there are different information available regards to the proofreading. So it is not exactly known whether the enzyme has any properties of proofreading or not. So let's talk about the RNA polymerase enzyme, which is important during the transcription process. So RNA polymerase is a DNA-dependent enzyme which synthesizes new strand in 5' to 3' direction. So in prokaryotes, the RNA polymerase enzyme consists of core enzyme, which involves 5 subunits. So this is the 5 subunit, and this 5 subunits includes 2 alpha. This is actually alpha, therefore it is 2 alpha, beta prime, and omega. And alpha subunit is important for enzyme assembly. Beta prime is important for binding of enzyme to the template strand, and beta subunit is important for 5' 3' RNA polymerase activity to synthesize new RNA strand. And there is this sigma subunit, which is, you can see here that it is present here, which is also not part of the core enzyme. So sigma subunit is very important in prokaryotic transcription process because it enables the RNA polymerase to recognize the promoter region on the DNA strand. So core enzyme itself cannot recognize the promoter region on DNA strand to start synthesizing new strand. So sigma subunit is critical and enables the RNA polymerase to recognize the promoter region on the DNA strand. And eventually this sigma subunit plus the core enzyme, it forms the RNA polymerase holoenzyme. So this is important that the RNA polymerase enzyme consists of this core enzyme as well as this sigma subunit, and eventually forms this RNA polymerase holoenzyme. And the another important information to know that RNA polymerase holoenzyme synthesizes all forms of RNA, which includes mRNA, rRNA, and tRNA. So now let's talk about the eukaryotes. So in eukaryotes, there are 3 different RNA polymerase enzymes. So we have RNA polymerase 1, which synthesizes rRNA, that is 45 subunit rRNA, and then which will then subsequently cleaved into different subunits. Then we have RNA polymerase 2, which synthesizes mRNA, and it also synthesizes some forms of small amount of small nuclear RNA as well as microRNA. And RNA polymerase 3, which synthesizes small RNA such as transfer RNA and 5S ribosomal RNA. It also synthesizes small nuclear RNA as well as small cytoplasmic RNA. Now, the one important thing to remember in eukaryotes that this all 3 RNA polymerase enzyme cannot just go and bind to the promoter region on the DNA strand. It also requires one specific protein which is called transcription factor. So transcription factor, basically it forms a DNA and protein complex on the promoter region, and then eventually that will result in the recruitment of these different RNA polymerase enzyme to initiate the transcription process. So just like we saw that how sigma subunit is important for which enables the RNA polymerase to recognize the promoter region on the DNA strand in prokaryotes, same way in eukaryotes it is transcription factor which is important to initiate the transcription process. So now I just want to quickly talk about the template and the non-template strand. So this is our DNA molecule, and of course this is a double-stranded DNA molecule, and let's just consider that the bottom strand is the template strand, which is also called as non-coding or antisense strand. And because it's a template strand, it will be used as a template to synthesize mRNA. And the top strand on the DNA strand is called non-template strand and coding or sense strand. And if you see, pay attention to the nucleotide sequence on the mRNA. So the nucleotide sequence on the mRNA are similar to the nucleotide sequence on the non-template strand, except the one difference is that mRNA has uracil instead of thymine, which is present on non-template strand. Because mRNA is a ribonucleotide, so we have here uracil instead of thymine. So that is the only difference. So now before we talk about the transcription process, I just want to quickly touch base on the structural organization of a gene. And one very important information I want to share, that whenever we mention about the DNA sequence, these are this nucleotide sequence in the promoter region or nucleotide sequence either on the upstream regulatory sequences or some nucleotide sequence on the control elements. Whenever you talk about the nucleotide sequence on the transcription, they are always written in 5' to 3' direction using the non-template strand. So this is something very important. And whereas on the other hand, RNA polymerase enzyme reads the template strand in the 3' to 5' direction to synthesize mRNA. So this is something very basic information, but it is very important when we talk about transcription. So now let's talk about the organization of a gene. So here we have a double-stranded DNA molecule, and the top strand is the non-template strand and the bottom strand is the template strand. So the site on the DNA molecule where the first ribonucleotide is added is given a number which is called +1 base, and this +1 base is also called as transcription start site, because from here the first ribonucleotide is added to initiate synthesizing the mRNA strand. Now the nucleotide sequence which comes before the transcription start site, they are given negative numbers, and they are also present towards the 5' end of the non-template strand, and they are also called as upstream of the transcription start site. Whereas the nucleotide sequence, which comes after the transcription start site, they are given the positive numbers, and they are also present towards the 3' end of the non-template strand, as well as they are also referred as downstream of transcription start site. So now nucleotide sequence which are present before the transcription start site, so specifically the one which are highlighted here in pink color, they are the sequence which are called promoters. And these promoters are basically, they are also called as recognition sequence, because this is the sequence which is going to be detected by either transcription factor or the sigma subunit. So it could be, it will be eventually detected by an enzyme or the protein to initiate the transcription. And nucleotide sequence on the right, which comes after the transcription start site here, which is highlighted in yellow, it is called as transcribed region. It is called as transcribed region because this is the region which will be eventually transcribed into mRNA. So this is basically the basic structural organization of a gene. So now let's talk about the transcription process in prokaryotes. So this is our structure of a prokaryotic gene, and this is our first promoter region, which is called as negative 35 sequence. And it is called as negative 35 sequence because it is present before the transcription start site at the negative 35 base. And there is another promoter which is also called as pribnow box, and the pribnow box is present on the negative 10 base before the transcription start site. And you can also see that the pribnow box is rich in adenine and thymine. So this is our to initiate the transcription process. Here we have RNA polymerase holoenzyme, and RNA polymerase holoenzyme detects this negative 35 sequence on the DNA strand, and which creates this closed complex within the DNA strand. And subsequently, the RNA polymerase holoenzyme moves down to the DNA strand and recognizes the pribnow box promoter region and binds to this promoter and creates an open complex. When I say open complex, means that it will start unwinding this 2 DNA strand by melting the hydrogen bond between thymine and adenine and result in this open complex. And also it is important to know that means the RNA polymerase holoenzyme performs the helicase activity to unwind this 2 DNA strand. And just like we learned in the replication process, that it is both thymine and adenine are paired together by 2 hydrogen bond, so it is easy or it requires less energy to break 2 hydrogen bonds between adenine and thymine, opposed to the 3 hydrogen bonds between guanine and cytosine. And therefore you can see that the pribnow box is rich in adenine and thymine because it can be easily the hydrogen bonds can be easily melt down. So when it creates this open complex, it creates this transcription bubble. So this open complex is known as transcription bubble, which is also very similar to the replication bubble we saw in DNA replication process. And this is our +1 base, which is also the transcription start site. And while RNA polymerase enzyme unwinding these 2 DNA strands, it also creates this torsional strain between this DNA strand, and which can result in the either in the positive or the negative supercoiling, and this supercoiling is inhibited by the enzyme called topoisomerase. And in prokaryotes, this topoisomerase enzyme is called also called as DNA gyrase. So this is basically the initiation of transcript, not the initiation but the recognition of the promoter region by RNA polymerase holoenzyme process. So now once we know this, now we can talk about how the new RNA strand is being synthesized with RNA polymerase holoenzyme. So here we have our open complex DNA strand. So now, for example, I have given this a DNA strand, which I'm going to use this as a template strand. So first thing, before we even learn about synthesizing new strand, couple of things we need to know, that RNA polymerase enzyme reads the template strand in 3 to 5 prime direction and synthesize the new strand in 5 prime to 3 prime direction. And second thing, we need to know that RNA polymerase enzyme does not require RNA primer to start synthesizing new strand, which is again opposed to the DNA polymerase enzyme, which requires RNA primer to synthesize new DNA strand. So now consider that this first nucleotide here is present at +1 base, that means it is present at the transcription start site. So RNA polymerase start adding the nucleotide from the transcription start site. So here RNA polymerase adds the nucleoside triphosphate. So it is nucleoside triphosphate. It is nucleoside because nucleoside is equal to sugar molecule, and here the sugar molecule is ribose sugar. So sugar molecule plus the nitrogenous base is equal to nucleoside, and triphosphate, and therefore it is called nucleoside triphosphate. And it is triphosphate because this nucleoside brings its own energy in the form of triphosphate, which will be then utilized to form the phosphodiester bond. And again, this nitrogenous base is complementary to the nitrogenous base on the DNA template strand. So now this is our +2 base, which is located next to the transcription start site. So RNA polymerase add the complementary nucleoside triphosphate, and it will then cleave this 2-phosphate group, which is called as pyrophosphate, and then this pyrophosphate is then converted into 2 orthophosphates. And these 2 orthophosphates are basically the powerhouse of the energy, which generate the energy, and this energy is then utilized to form the phosphodiester bond between 3' hydroxyl group of one nucleotide and 5' phosphate group of the other nucleotide. So similarly, RNA polymerase add the nucleotide, which is, and in this case it is uracil, because you need to remember that these are ribonucleotides. These are therefore it is uracil and not the thymine, and which is complementary to the adenine on the DNA strand. So it adds this nucleoside triphosphate here, cleaves the pyrophosphate, generate the energy to form the phosphodiester bond. Similarly, it again brings the nucleoside triphosphate, cleaves the phosphate pyrophosphate, generate energy to synthesize the phosphodiester bond, and eventually it completes the formation of the small stretch of mRNA. So as you can see here on the left, So this is the RNA polymerase holoenzyme, which eventually synthesize this small stretch of mRNA. You can see that here, which also has this 3-phosphate group at the end. So you can see that there is a 3-phosphate group at the end, which is attached to the small RNA molecule. Now this small stretch of RNA molecule, which is composed of 9 to 12 nucleotides, these are called oligonucleotides. And once this oligonucleotide is formed, the sigma subunit on the holoenzyme is then released. And when this sigma subunit is released, it kind of indicates the completion of the transcription initiation process in prokaryotes. So once this is released, the RNA polymerase core enzyme is highly processive and goes on to synthesize the remaining of the mRNA strand. So it eventually completes the synthesis of mRNA strand in 5' to 3' direction. So far we saw that RNA polymerase holoenzyme recognize the promoter region, and then it unwind the DNA strand. So you can see that the DNA strand is unwind, and then eventually it synthesizes this small stretch of RNA. So these first initial steps, they are called initiation steps in the transcription process. And once the oligonucleotides are formed, the sigma subunit is released, and then the RNA polymerase core enzyme then synthesize the remainder of the mRNA strand in a very highly processive manner. That means it elongates the remaining of the mRNA strand, and therefore this is called the elongation process of transcription in prokaryotes. So now this is the transcription initiation and elongation in prokaryotes. So now let's talk about the transcription initiation in eukaryotes. And in eukaryotes, it is a little bit different than prokaryotes because the transcription process is more complex in eukaryotes compared to the transcription process in prokaryotes. So in eukaryotes, if you remember that the DNA molecule is packed into the chromosome. So this is our chromosome, which is basically the condensed form of chromatin. So this is the chromatin, and chromatin is basically composed of DNA and protein. So proteins here, they are majority of the time they are histone proteins. So this DNA is wrapped around the histone proteins, and when they are tightly packed, they form this condensed chromatin. So now chromatin is basically present in two different forms. When it is present in heterochromatin form, it is tightly packed and forms the condensed chromatin loop, and this form, this heterochromatin, is considered to be transcriptionally inactive. On the other hand, when chromatin is present in euchromatin form, the chromatin fibers are loosely packed, and they are considered to be easily accessible chromatin loop, and it is also considered to be transcriptionally active. And the interconversion between this heterochromatin to euchromatin and euchromatin to heterochromatin is called as chromatin remodeling. So in order for transcription process to occur, it is very important that the heterochromatin is converted into euchromatin in order for transcription process to occur. So let's see the mechanism of chromatin remodeling. So suppose this is the heterochromatin formation. So this blue, these are the histone protein, and this dark blue strings, they are DNA molecule, DNA strands, which are wrapped around the histone proteins. So on the N terminal of histone proteins, which is which has a high content of positively charged lysine residue. So this positive charge on the lysine residue forms the ionic bond with the negatively charged phosphate group present on the DNA strand. And when it creates this bond, it results into stronger interaction between the DNA strand and the histone protein, and create this heterochromatin formation, which is also the tightly or the condensed form of chromatin loop. So there is an enzyme which is known as histone acetyltransferase. So histone acetyltransferase acetylate the lysine residue on the histone protein and removes this positive charge on the lysine residue, which will then result into the reduction in the interaction between the histone protein and the DNA strand, and resulting in the formation of resulting in the euchromatin formation. So this is histone. So histone acetyltransferase is a very important protein which play a role in chromatin remodeling. On the other hand, there is an enzyme which is called as histone deacetylates. So it removes this acetyl group from the lysine residue, which result into the restoration of the positive group, and then eventually it will restore the stronger interaction between the histone and the DNA strand, which will result into heterochromatin formation. So these are two important enzymes which play a role in regulating the transcription process in eukaryotes. So now that we have learned about the chromatin remodeling, now we can talk about the transcription initiation in eukaryotes. So like I mentioned before, that initiation of eukaryotic transcription requires a specific protein which is called transcription factor. So there are different types of transcription factors, which includes transcription factor 2A, 2B, 2D, 2E, 2F, and 2H. And before we even go and talk about that the transcription factor more, I just want to quickly discuss the structural organization of a eukaryotic gene. So here we have three different promoter region. So the first promoter, which is called TATA box, which is present on the -25 base before the transcription start site, and you can see that it is rich in adenine and thymine. And there are basal transcription factor which binds to this TATA box. Now the second promoter region, which is called, I call it as a cat box. So this cat box promoter region is present on the -80 base before the transcription start site, and the general transcription factors which recognize and binds on the cat box. And lastly, there is also a promoter region which is called GC box, which is present on -100 base of before the transcription start site, and it is rich in guanine and cytosine. So for transcription process to initiate in eukaryotes, the transcription factor 2D protein, which recognize the TATA box promoter region and binds on the TATA box, and the TATA box promoter region is also called the core promoter. So it binds to the TATA box on the core promoter, and transcription factor 2A, which promote the stability of the interaction between transcription factor 2D on the TATA box promoter region. And the binding of transcription factor 2D to TATA box promoter is very crucial, because this is followed by the recruitment of other transcription factors. So the binding of this is very crucial. So once this binds on the TATA box, it recruits the transcription factor 2H, and which will perform the helicase activity to melt the DNA strand and start unwinding the DNA strand on the TATA box promoter region. And it also recruits transcription factor 2F, which also brings along the RNA polymerase 2 enzyme on the transcription start site, and transcription factor 2B, which helps accurately position the RNA polymerase 2 enzyme on the transcription start site to initiate synthesizing new RNA strand. So the synthesis of the new RNA strand is not different than synthesis of the RNA strand we saw in prokaryotes. So RNA polymerase 2 enzyme, basically it initially synthesizes this small stretch of RNA, and then it goes on to synthesize to complete the synthesis of the remainder of the RNA strand, which is actually called pre-mRNA. It is different than prokaryotes because it is called pre-mRNA because it requires some modification, and therefore it requires some modification to finally generate the mRNA strand, and therefore it is called pre-mRNA. And that modification process is called the post-transcriptional modification of RNA, and we will be talking about that in our subsequent video. So don't worry about that. Do not pay a lot of attention that what is that post-transcriptional modification, but I will definitely be talking about that in the subsequent videos. So this is the initial process where the RNA polymerase, where the transcription factor recognize and binds to the promoter region, and then recruit the other transcription factor to create this transcription bubble, and then as well as it also recruits the RNA polymerase 2 enzyme, and then start synthesizing the to initiate the synthesis of RNA strand. So that initial process is the transcription initiation steps, whereas when the RNA polymerase enzyme complete synthesizing the long stretch of RNA, that is the elongation process of transcription in eukaryotes. So lastly, I just want to quickly mention about the importance of RNA polymerase 2 enzyme. So there is a condition called alpha-amanitin poisoning, and alpha-amanitin is a potent toxic chemical which is produced by poisonous mushroom known as Amanita phalloides, which is also called as death cap, and you will see why it is called as death cap. So alpha-amanitin is basically, it inhibits this RNA polymerase 2 enzyme. So if the RNA polymerase 2 enzyme is inhibited, the synthesis of mRNA is inhibited, and if the synthesis of mRNA is inhibited, it will eventually inhibit the synthesis of a protein. So it inhibits both mRNA and protein synthesis, and this is found to be extremely hepatotoxic to humans as well as domestic pets which consume this wild mushroom which contains this alpha-amanitin toxic chemical. And the symptoms of alpha-amanitin poisoning includes cramp, diarrhea, and then eventually it can lead to renal failure, liver failure, and then coma, and eventually death. And that's why it is called as death cap, because it is extremely lethal who consume this wild mushroom. So here guys, this is the initiation and elongation process of transcription in both prokaryotes and eukaryotes, and I will also be uploading the video on termination of transcription as well as the post-transcriptional modification of RNA. So I really hope that you enjoyed this video, and most importantly you learn something new from this video. And if you do so, please like, share and share the video and subscribe the channel. Thanks so much, and I'll see you soon.